Now let's revise and conclude our last lecture of DESeq2. So DESeq2 is a powerful and widely used R package for the differential gene expression analysis based on our RNA sequencing data. And the analysis pipeline that I'm going to show you, we can use that to import, pre-process the count data, and then explore the data analysis, and using a differential expression we can visualize the result. So let's move on to that. So we are going to open our R window, and opening the R window, opening our R studio, we are going to use the R studio for our differential expression analysis. So let's just wait while the R is loading, and then we are going to use our operating system. Yes. So here we are. Then I'm going to close this tab, and then I'm going to open a new file in my folder, which is in our coding section, DESeq2. So I'm going to open the two file, which is get data and run the DESeq2. So in the get data, I have created a which is a directory, and also an airway library, which I'm going to download the sample data. So here you can run this command to get the data, and I already have the sample data here. I'm going to show you, these are the sample information. Now here you can see the RNA-Seq data which I was talking about. So here are the ensemble genes, ensemble gene IDs, which you can visualize these. And here is the expression data. So you can get the idea of how these data packages are represented. So here is the unwanted column, and there are no sample on it, and I'm gonna remove it. I have removed the unsampled data, and now you can see I'm going to remove that as well. Here is the data line. Here is the gene IDs, and here are the sample data. So I'm going to save this Excel file, and then move on to our operating environment. And then after getting the data, you need to close this command and then move on. So here is the script for the differential gene expression analysis using the DESeq2 package. First of all, you have to set the directory. For example, I'm going to copy this and then paste it here. The one thing you have to manage is you have to remove these slashes and bring out the forward slash. You have to change this, and then the set directory, you can also set your directory. After that, we are going to load the libraries. So DESeq2, tidyverse, and airway are the three libraries that I used. And then we are going to prepare the count data. Here you can read the count data. I have read the Excel file. So let's just run the code with it. So as I am explaining. So here you can see our code is running, and then let's move to the tidyverse, and let's load the tidyverse package, and then load the airway library, and then read the count data, and then show us how your data is looking right now. Here you can see our data is being loaded. Then we can read the sample info. Here we can going to read the sample info. All the sample info are present there, and then move on to construct a DESeq object. And then we are going to run the command of constructing the error in summarize. Let's just ignore this error, and then move on to the DDS object. Error DDS not found. So there is a problem with this. I think we have manipulated that data which we were not supposed to. So just going to save that. And yes, we are going to replace it and move to the R session, and then read the counts data, and then read the sample info, and then it is saying that DDS was not on. So there is a problem with our DESeq2 package. So let's load this again. If it still gives us the data TDS was not found. So I think there is a problem with our DESeq package, running the DESeq package. So let's move to our folder where we were getting the data and here. So sorry for that. So here we can see if I'm gonna close this and open the run DESeq2. And again, we are going to all my libraries are loading well and the data is currently loading. So warning, in the column name, row's name, the longer object is not multiple for the shorter length. So here you can see error in the DESeq from the matrix was not true. So DDS was not found. So the DDS object is not picking up. So we have to close this R session and then restart again. If these main problems that I have gone through are caused by the package that you are using is causing this problem. For example, there is a new update of DESeq2 package that is available and we have not updated that yet, then it's going to cause a problem as well. So I already knew there will be a problem of DESeq, so I'm just going to show you how to manage with that. So we are going to set the directory again and then load the libraries, and then we are going to see if it's going to say that there is any kind of warning that we are not getting any kind of data. So if we again install our DESeq2, let's just say we move to the packages here and then search for DESeq2 differential gene expression, and then uncheck this, and then again check this, and then see what happens. So move to the tidyverse and then move to the airway library and then the counts data. Then again load our counts data and then read the column table and then this. And there is a package and there is error DTS was not found. So I'm going to update the package of DESeq2. So in this video, there was a problem with our DESeq2. So there is a lot of updates that are already being represented here. So let's just uninstall the DESeq2 package and again install it. BioC manager install DESeq package. You can see the DESeq package. And okay, so I have not used this calling. So it is installing our DESeq2 package and we're gonna see if the DESeq2 works or not. So let's just wait while the DESeq2 is being installed again. So when you're working with the R, there are going to be a lot of problems that you are going to face, just like this. And last night I was running this package and it was already going through well and all this stuff. But this problem is caused by when a newer version of package is available. So installing package is in use, warning DESeq2 is in use and will not be installed. Installing path are not writable, unable to write the packages. So it is saying that if I want to update the several other packages that are available, so I'm going to cancel this file and make a new one, and then we are going to install our DESeq2 package as well. File C manager install, and then I'm gonna type our DESeq2 again here and here. And while the other packages are being unpacked and then installed correctly. So while we are waiting, let's talk about the RNA sequencing data. So what basically is RNA sequencing data that is being represented here? So RNA sequencing data is basically our re- I'm gonna show you a database where we can get this type of data, NCBI SRA. So SRA is the archive sequence archive that is available for the multiple cloud operated and NCBI servers, which we can use to store the raw sequencing data and alignment information of our data analysis. So how you can get your data? So just type human RNA-Seq data. I'm sure it's gonna give us some SRA files which we can use. So here you're seeing it is treated with AMG compound. You can get this and then you can analyze this. So why we are using the RNA-Seq data and why we are dealing with that? Because now that RNA-Seq is currently being used for a lot of other analysis, so that's why we are using this kind of data. So I'm gonna copy this command and then close this file as well, and then restart the R session as well, and then let's see how, if our problem is solved or not. So here we are again in our R studio while it's opening up. I don't know why it's loads so late, because this version of R is not quite so stable because I'm getting the errors right away. So let's make a new window and then here paste our DESeq2 information and then install our DESeq2. So let's just wait while the DESeq2 is installing. So while we're waiting, I'm going to tell you about the bioconductor. So bioconductor is the installer that we use in R for the bioinformatics analysis, like bioinformatics packages, which is DESeq2 is also available in bioconductor. Here we can search for the DESeq2, DESeq2. So you can see bioconductor is hosting these DESeq2 data differential gene expression. So here you can see they are giving us the command of how to install the DESeq2 package. So it is being downloaded to 100%, and then DESeq2 is successfully unpacked, and it is saying to update the packages as well. So let's just update all the packages that are available for updates. So now just we are going to open our run DESeq2 file. So all these files will be available to your my GitHub account. You can get these codes and modify them according to your need and then get the analysis done. So okay, so we should wait while it's updating. So while they are updating our interface, we have to wait and then run the commands again. So while now you are seeing that DESeq2 is currently loading. So now we are going to run again this command. So let's just set that directory and then load the libraries of DESeq2, load libraries of tidyverse, and I'm seeing a lot of error in tidyverse as well, because tidyverse, I think tidyverse update is already available. So let's just go with it if we can get that data again. So there is a issue with the data, I think so. Error in the DESeq format column data is not true. So we are going to again get the data. So get data, we are going to open the get data file and then set that directory. After that, load the directory, load data airways, and then open this, and then sample info, load these sample files, and then get the comps data. So here in our folder, let's just read the sample and counts data, and then again run our get data file. Here we can see we are going to get this file, and then we can see yes, we get the count data again. And now in our R session, we can use these libraries to see if that data is currently working. So here you can see now there is no error at all. It is saying true, and the DESeq2 object is now loading some variable designs. So they are converting to factors. Now you are seeing that metadata is also already available, and there are objects that are creating here. So DDS, we are going to open that DDS file. Now let's just, so there is steps that already mentioned in our previous lecture. Now we see we are going to create our the DDS metrics, and and then open the files and then see if we can get that gene expression. So it is estimating our size factor, dispersion estimates, mean dispersion relationship, and final dispersion, and then fitting the model and then testing the model. So now we can get the results of DDS. Here you can see there will be a file named results as well. Here is a DDS file, and then open this. Now here you are seeing all the values are operated, and then we are going to explore the results. So after exploring the results, here we can move on to plotting this information, and then we are going to plot our volcano plot that we can appear here. Here is the mean of normalized count, and here is the mean of fortune, and then we can open this image in a new session. Here you can see the genes that are represented, and these blue kind of values are our highest FPKM values that. So I think you have seen a lot of this data in other tutorials as well. So that was it for the DESeq2. I'm going to give this code to the my GitHub account, and then you can use this for your analysis as well. So that was it for